Okay, hi, my name is Sandra Healy and I'm the Programme Manager for TRAIN-ERS. Now TRAIN-ERS is it's a European Horizon 2020 funded project. It's an innovative training network. So this training network is going to focus really on investigating endoplasmic reticulum stress in health and disease. But before I actually go on and talk about the science, I just want to explain a little bit about the programme. So basically, this program is a training network for 14 different PhD students coming from a wide number of countries. And it's got three main elements. So these students are going to spend 3 or 4 years carrying out research projects. But in addition to that, they're also going to there's a comprehensive program to give them scientific skills and give them transferable skills. And in addition to that, then, the students will also have the opportunity to go on secondments. So secondments to different countries, secondments to industry and to hospitals, to give them a good experience besides just the academic setting. And in addition, they'll have the opportunity to go to network-wide meetings of all the scientists involved in this program every 6 months. So they'll meet and and network with all these scientists. But probably most importantly, from our point of view, this program will really increase our understanding of the endoplasmic reticulum stress response and how we can exploit our knowledge of that for therapeutic purposes. So the endoplasmic reticulum, okay, it's a mouthful. I'm going to call it the ER, okay, for short. So what is it? Well, it's a subcellular organelle, which essentially means it's a compartment within the cell. So if we look at the structure of the cell here, you can see we have the cell membrane surrounding the cell. And then within the cell, there are different compartments. And the different compartments have specialized functions. So for example, in the center here, we have the nucleus. So the nucleus is essentially the command and control center of the cell. We have these blue things here. They're called mitochondria. And these are sort of like power stations in the cell, generating all the energy that the cell needs. We have the green ones here are lysosomes. They're essentially kind of garbage disposal and recycling centers. So the endoplasmic reticulum is this structure here that you can see around the nucleus. So it's this network of tubules. And if you actually look at it under an electron microscope, this is what you see. So here we have the nucleus, and here you have all this network of tubules. If we focus in on it a little bit more, you can see these are all the tubules, and they have a kind of a rough appearance with these little dots on them. And what these little dots are, they're actually protein manufacturing complexes that are called ribosomes. So if you look at the kind of cartoon image of the ER here, these are our tubules, and all these little dots on it are the protein manufacturing complexes that are called ribosomes. And really one of the main function of the ER is to make new proteins. So these are new proteins that are they're proteins that are going to be exported from the cell, or else they're proteins that are only found in the membranes of the cell. So it's very specialized, only proteins for export or only proteins found in cell membranes. So if we just look, okay, we're going into the science here now. So if we look to see how proteins are actually made in the ER, so here we have this kind of purple line here is the genetic material that has the instructions on how to make a new protein. Then we have the ribosome, which is our protein manufacturing complex, and this basically reads the instructions on the genetic material and translates it into a protein. So you can see that as it moves along the genetic material, it starts to make the protein that you can see here. Now the first part of the protein is essentially a kind of a postcode, and it says, okay, this protein has to go to a certain part of the cell. So if we have the postcode here is saying this protein needs to be exported from the cell. So this little postcode will bring the protein complex to the endoplasmic reticulum, and then as the ribosome reads the genetic material, it translates it into a protein, and the protein is inserted into the ER. Okay, so what we're left with in the end is this mature protein in the ER. So in the ER then, so the proteins are made, but they also have to be folded properly, and they also have some modifications done to them. So they have things like sugar groups added to them. So once the sugar groups and the modifications are carried out, then we want to export the protein from the cell. So what happens is that the protein gets kind of put into these vesicles that bud off from the ER, And the vesicles then travel to the cell membrane, and they fuse with the cell membrane, and then the protein is released outside the cell. Okay, so the protein is exported or secreted. Now, protein folding is extremely important. If a protein isn't folded properly, it won't be able to function properly. So it has to attain a specific three-dimensional shape in order to be active and to carry out its job. And if proteins are misfolded or unfolded, it means they're not going to function properly. And not only that, they also tend to be very sticky, and they stick to each other and form these big aggregates, which is very bad for the cell. So why is the ER important? Well, first of all, all cells need proteins for the membranes, and they need to export proteins. And the average cell, about 30% of the proteins that it produces are made in the ER. However, there are some cells that really make huge amounts of protein, and these are specialized secretory cells. So, for example, cells in your digestive system and in your mouth make digestive enzymes to allow you to break down your food. There are blood cells that make antibodies to allow you to fight infection. You also have specialized cells in the pancreas that secrete insulin that allows you to regulate your blood glucose levels. So all these proteins are exported, but they have to be folded properly first. And in order for that to happen, the ER has a very rigorous quality control system. Okay, and so the ER will recognize if a protein is not folded correctly, and it gets rid of it, okay? It destroys it. And this is a very complex, energy-consuming process. And if you just look here at this, so we have a newly synthesized protein, and there are enzymes inside in the ER that help the protein to fold properly. If it's folded properly, then as we've seen, it gets packaged into vesicles and exported. If it's not folded properly, this is recognized, and the protein is sent to be destroyed instead. So the ER is important, okay, in every cell type in the body, but it's especially important, has a very important physiological role in the bone marrow, in the liver, in the gastrointestinal system, in the pancreas, and the brain. So the ER makes proteins, but the ER is actually very susceptible to stress. Now, there are lots of different things that can stress the ER. So, for example, changes in pH, low oxygen levels. If you have mutated proteins that are difficult to fold because they have mutations, glucose, low levels of glucose means that the cell will have low energy levels, so it won't be able to fold the proteins properly. Also, very high levels of glucose are toxic and stress the ER. High levels of fat, viral infection, and calcium imbalance. So all these factors can stress the ER. And when the ER is stressed, okay, it's not able to fold the proteins properly anymore. So what happens is you get accumulating unfolded proteins. And this isn't good for the cell, okay? It's bad. So the cell basically thinks, oh, okay, this is not good. So it activates a stress response that's called the unfolded protein response, or UPR. So essentially, what this unfolded protein response is activated when you have all these unfolded proteins accumulating. And it says, okay, it says to the cell, we need to stop making new proteins, okay, until we can get rid of all these unfolded proteins. We also need to try and increase our capacity to fold proteins, and we need to make sure that we get rid of all the unfolded proteins. So basically what the unfolded protein response, or UPR, does is it allows cells to cope with ER stress. So the UPR then will allow cells to cope with mild or transient ER stress. So it activates molecular pathways that allow the cell to survive and adapt. However, if the ER stress is very severe, or if it's prolonged, so it happens over a long period of time, there's too much damage to the cell. There's too many unfolded proteins accumulating. So the UPR then instead activates a different arm, activates molecular pathways that lead to cell death. Okay, so the UPR then has two arms. One is activated in response to mild stress, and it allows the cells to adapt and survive. The other is activated when the stress is very severe, and it causes the cells to die. Okay, so we've seen then that the unfolded protein response is activated when you have ER stress. But recently, scientists have found that the UPR is very tightly connected to both metabolic processes and to inflammatory processes. So when you have activation of the UPR, you also alter metabolic responses and you alter inflammatory responses. And because of that, then, the ER stress and the UPR has been shown to contribute to the development of many different diseases. So we know that ER stress and the UPR is involved in metabolic diseases such as obesity, arteriosclerosis, cardiovascular disease, and so on. We also know that it's involved in the development of inflammatory disease such as Crohn's disease, rheumatoid arthritis, type 1 diabetes. It's been known for a long time that neurodegenerative diseases such as ALS, Parkinson's, Alzheimer's are all characterized by protein misfolding. And also, as Eilish pointed out, there's a connection between the inflammatory response, okay, and the development of neurodegenerative diseases. And then, in addition, cancer cells are very cunning. So what they do is you have the two arms of the UPR. So what the cancer cells do is they try to turn off the death arm of the UPR and keep the survival arm of the UPR. So that allows the cancer cells then to grow in environments where normal cells wouldn't be able to grow. So ER stress and UPR isn't the cause of all these diseases, but what it is it's contributing to the development of these diseases. So our theory is that if we can find drugs that can modulate the unfolded protein response, they might be useful for treating many of these diseases. Okay, so TRAIN-ERS then has three main aims. The first one is really to try and understand how cells respond to ER stress. Look at the effects of ER stress on the cells and how the cells actually respond to it. The next one is to understand how ER stress and the unfolded protein response actually contribute to the development of diseases. And then the third thing is to use this understanding here to try and develop new drugs for a wide range of diseases. So these three aims, okay, are addressed by three work packages in TRAIN-ERS. So the first work package will be looking at ER stress biology. So basically this work package is aiming to understand the effects of ER stress on cells and how the cells respond at the molecular level. Okay, so this just kind of gives you an overview of some of what we know at the moment. So when you have unfolded proteins accumulating in the ER, that's sensed by three proteins here in the ER. So they sense this accumulation of unfolded proteins, and they activate these molecular pathways. So what we want to know is, what are these pathways, and what are the key players in these pathways? So if we know what the key players in the pathways are, we might be able to alter them with drugs, and that might allow us to actually alter the balance between whether cells survive or cells die. In addition, we want to understand better how the UPR communicates with the inflammatory and the metabolic processes. The second work package will be looking at ER stress and disease, and one of the main diseases that we're going to be looking at is cancer. So we know that cancer cells are under constant and very high levels of ER stress. That's because cancer cells tend to grow very quickly, so they need a high rate of protein production to make new membranes and so on. So they have stress from the high demand on the ER for new proteins. Also, tumours, at least at the beginning, often have an inadequate blood supply. So because of that, then they tend to be lacking nutrients and oxygen, and that also increases the ER stress on these cells. And then finally, cancers often have mutated proteins that are difficult to fold. So all these contribute to really making having a high level of ER stress on the cancer cells. Now, normal cells would die with that level of ER stress. However, cancer cells have found ways to try turning off the death arm of the UPR, and instead they keep the survival arm activated all the time. And that allows them to grow and survive in environments where normal cells can't. And not only does it allow them to survive and to grow, but it also means that because they've upregulated these survival arms, it also means that they're more resistant to chemotherapeutic drugs. Another disease that we want to look at, other class of diseases we want to look at, are neurodegenerative diseases. So we know that the UPR is needed for normal cells, normal nerve cells, okay? So normal nerve cells are exposed to ER stress every day, and they need the UPR to help them to cope with that ER stress. And we also know that protein misfolding is very common in neurodegenerative diseases, and that there's a close association between neurodegeneration and the upregulation of ER stress markers. Now, the neurodegenerative disease that we're concentrating on is called amyotrophic lateral sclerosis, or ALS. Now, you might have heard it referred to as Lou Gehrig's disease. So Lou Gehrig was a baseball player in the 1920s in America. He played for the New York Yankees. He played with the likes of Babe Ruth and Joe DiMaggio. And he helped New York Yankees win 7 World Series championships. So he developed this disease, so it's often referred to as Lou Gehrig's disease. And what it is it's a disease of progressive paralysis. And that's because of the death of motor neuron cells in the brain and the spinal cord. Now, many different mutations have been associated with ALS, and most of these mutations tend to cause protein misfolding and aggregation. So just an example is shown here. We have this protein SOD1, when it's mutated, has been shown to lead to ALS. Now, SOD1, as you can see, is actually not inside in the ER at all. It's outside the ER. But when it's mutated, it doesn't fold properly, and it's sticky. And it sticks to a protein that is in the ER membrane here. Now, this protein here is required to get rid of unfolded proteins. Okay, so normally it gets rid of the unfolded proteins and everything's fine. However, if you have a mutant SOD1 here, it binds to this and stops it acting. So this now can no longer get rid of unfolded proteins. The unfolded proteins accumulate to high levels, activating the death arm of the UPR, and you get loss of cell viability. Okay. So our last work package then is to try to apply our understanding to develop new drugs to treat a wide range of diseases. So, for example, for degenerative diseases, neurodegeneration, also things like diabetes, where you have death of the beta cells in the pancreas, we want to try and prevent cell death. Okay, and there's 2 ways that we can go about that. We'll try to develop drugs that can enhance the cell survival arm of the UPR. And in addition, we'll try to find other drugs or other ways of reducing the ER stress on these cells. The other diseases are cancers. So in cancers, we want to do the opposite. So we want to enhance the cell death in cancers. So we want to, there's 2 ways of going about that. We can try to block the survival arm. So we know the cancers are relying on this survival arm. So if we can find drugs to block that, and at the same time, enhance the ER stress on these cells so that cells are forced to die. Okay, so so TRAIN-ERS is that we have 11 partners. It's a 3.6 million funded project funded by the EU, and you can see that the red dots here show where the partners are based around Europe, and we have one in Israel. There'll be 14 PhD students. And then in addition, we have these 6 partner organizations here that are involved in secondments and training. The project is coordinated by Professor Afshin Somali here at NUI Galway. And really, I hope I've given you some enlightenment as to what endoplasmic reticulum is and how maybe we can exploit our knowledge of this for future drug development to treat a wide range of diseases. Okay, and I haven't sent you to sleep. Okay, thank you very much.